alaikum. Hello everyone. My name is Hoor Ameen, a fourth year medical student. In this video, I'm going to explain a section of the central nervous system chapter, which is the brain protection section. So we will start here. So the brain is protected by bone, which is the skull, meninges, cerebrospinal fluid, and the blood-brain barrier. We will be discussing each of these in the following slides. We will skip the bone for now. We will start with the meninges. Now the meninges are three connective tissue membranes that lie just external to the CNS organs. We will zoom in here. Okay, so we have three important layers of the meninges, which are the dura mater, the arachnoid mater, and the pia mater. These are connective tissue. The dura mater, as you can see here below the bone, it has two important layers, which are the periosteal layer, and as the name indicates, it's below the bone or surrounding the bone. So this is periosteal, and we have the meningeal layer. We will discuss them in details. Then we have below them the arachnoid mater here and the pia mater. Now these meninges are important because, as you can see here, they cover the CNS organs, which are the brain and the spinal cord. They also cover the blood vessels and the venous sinuses within the brain, and they contain the CSF or the cerebrospinal fluid, as we will discuss in the next slides. Also, they form partitions. So as you can see here, these are some partitions formed within the organs of the CNS, and we will see after. Okay, so as a mnemonic to remember the layers from the inside to the outside, I use this mnemonic. So the membranes act as a pad that protects the brain. So a pad from interior to exterior. P stands for pia mater, A for arachnoid mater, and D for dura mater. Okay. We will start with dura mater, which is the outermost layer. Now this is a tough layer, and I remember the dura mater as a rule of twos. So you will see that we have in this layer, we have two layered sheets, two layers, we have two unique structures, and two important spaces. Okay, so it's a rule of twos. Now these two layers are the periosteal layer, as we saw, and the meningeal layer. The periosteal is the superficial layer that attaches to the inner surface of the skull, the periosteum. And the meningeal layer is the deeper layer that forms the true external covering of the brain and continues caudally in the vertebral canal as the spinal dura mater. What does that mean? Again, in this picture, you can see that this is the periosteal layer. It's adherent to the bone. Okay, it is attached to the bone. At some spaces here, the meningeal layer, you can see both these layers are attached to each other, but at some points they separate. So the meningeal layer can go down through the organ and form here a partition. So here it separates, it forms a cavity between the periosteal and the meningeal layer, and then it attaches again, they attach again. At this cavity, there will be venous blood. So a cavity or a sinus will be formed at the site of separation, which will be filled by venous blood. So this will be called a dural venous sinus. And here where the meningeal layer goes down, it will form a partition or a septa that divides the organ or the brain into two hemispheres. So here you can see that we have two important layers and two unique structures formed by the dura mater. These are the dural sinuses at the point of separations and the dural partitions formed only by the meningeal layer. So as you can see here, the periosteal layer doesn't go down. It's only the meningeal layer that moves caudally. That's why it's the only layer that will protect or will cover the spinal cord. So the spinal cord is not covered by the periosteal layer, it's just the meningeal layer of the dura mater. Alright? So here, as we said, the two layers separate at some points to create two unique structures, which are the dural venous sinuses and the dural septi or partitions. And there are two important spaces within this layer or surrounding this layer. These are the epidural space or extradural space, which is just above the periosteal layer, and we have the subdural space, which is just below the meningeal layer. This is the subdural space. Hi, the subdural space. So these spaces are important because sometimes trauma happens and leads to hemorrhage within these spaces. Okay. Now, we mentioned dural septi or dural partitions that are formed by the meningeal layer of the dura mater. What's the importance of these septi? Why are they formed? Now, these septi will limit the excessive movement of the brain within the cranium. يعني كأنه موجودين كدعائم للدماغ حتى ما يتحرك بشكل كبير, to limit the movement. Now, what are these dural septi? We have three important septi to be remembered. The first one is the falx cerebri. Falx means a C-shaped structure or a sickle-shaped structure. Cerebri pertaining to the cerebrum اللي هو the brain here. هذا cerebrum. So falx cerebri is a C-shaped fold that will divide the cerebral hemispheres. So it's just at the middle of the cerebral hemispheres here. This is the falx cerebri. You can see here the brain. It has two cerebral hemispheres, and in the middle we have a fissure in between called the longitudinal fissure. So here is exactly where the falx cerebri lies. Okay. So it dips into the longitudinal fissure between the cerebral hemispheres, and anteriorly it attaches to the crista galli of the ethmoid bone. Here's the ethmoid bone. It's a bone of the skull, and it has a protrusion here called crista galli or crista galli. So it just attaches here and moves backwards. Now, as it moves backwards here, this is the falx cerebri, we will reach the cerebellum. So at the cerebellum, it will continue, the dura mater will continue here, but it will have another name. It will be the falx cerebelli, pertaining to the cerebellum. So here, as it ends here, it will continue as the falx cerebelli, which lies between the two hemispheres of the cerebellum. This is the cerebellum. It has two hemispheres, and they are divided by the vermis. So the vermis is just like the longitudinal fissure of the cerebrum, but it's called the vermis. So here exactly where the falx cerebelli lies. So it continuing inferiorly from the posterior falx cerebri here. This small midline partition runs along the vermis of the cerebellum. Okay, just at the middle of the cerebellum hemispheres. Lastly, we have the tentorium cerebelli. Tentorium, as the name indicates, it's like a tent above the cerebellum. So here's the cerebellum, and here's the cerebrum. Here is the tentorium cerebelli. So it's just above the cerebellum. It separates both the cerebrum and the cerebellum here. So it resembles a tent over the cerebellum. This nearly horizontal dural fold extends into the transverse fissure between the cerebral hemispheres and the cerebellum. So here, the transverse fissure in between, we will have the tentorium cerebelli. Remember, these are formed by the meningeal layer of the dura mater. Again, this is just a picture to show the dura mater. This is the periosteal layer that is adherent to the skull bone, and this is the meningeal layer that goes down to form the dural partition, and at the site of separation we will have the venous sinuses. Okay, now moving to the arachnoid matter. Arachnoid matter is the middle meninx, and it forms a loose brain covering, never dipping into the sulci at the cerebral surface. We will see. It is separated from the dura mater by a narrow serous cavity, which is the subdural space, which contains a film of fluid. I just showed you the subdural space here. The subdural space اللي هي just below the meningeal layer. This is the subdural space. Exactly below it, we will have the arachnoid matter. Now, beneath the arachnoid matter is the wide subarachnoid space. This is spider web-like extension. It has spider web-like extensions that span this space and secure the arachnoid matter to the underlying biomatter. What does that mean? Again, this is our arachnoid matter. Okay, it has some spider web-like extensions within this space. This space below the arachnoid matter is called the subarachnoid space, يعني below the arachnoid space. هاي spider web-like extensions will allow the arachnoid matter to adhere to the biomatter, to be attached to the biomatter, which is just adherent to the brain. Okay, لذلك احنا هاي بالعربي نسميها الأم العنكبوتية. It looks like شبكة العنكبوت. فراح تخلي الأم العنكبوتية هاي تلصق بالأم الحنون اللي هي biomatter that lies adherently to the brain. Now, this space is very, very, very important. Why? Because it contains a very important structure, or fluid, which is the cerebrospinal fluid. And it also contains the largest blood vessels that serve the brain. And because the arachnoid matter is fine and elastic, these blood vessels are poorly protected. See here, in this space, we have the CSF, cerebrospinal fluid, and these blood vessels, which are very important. And because it is not a tough layer, these blood vessels are liable to be injured. And sometimes injury happens, hemorrhage happens within this space, leading to subarachnoid hemorrhage, which is a life-threatening condition. So this is for the space. Again, we have projections of the arachnoid matter called the arachnoid granulations. These protrude superiorly through the dura mater and into the superior sagittal. Again, we will see them here. High arachnoid matter. You can see these granulations, these protrusions. They protruded through the dura mater here, and they reached the superior sagittal sinus, or the venous sinus here. This will have an important function that we will discuss, but just remember the structure here. Okay, so it has protrusions outside and spider web-like projections inside. Now, lastly, we have the biomatter, اللي هي gentle mother, الأم الحنون. Everything in this matter is gentle and cute, so it's a delicate connective tissue, and it contains tiny blood vessels. It acts like a plastic wrap. كأنه جبنا بلاستيك راب وغلفنا البرين بكل ضمير، يعني بحيث even within the fissures we have this biomatter. It covers all the fissures of the brain and all the convolutions. Alright, now this is for the meninges. A clinical correlation here: whenever you hear encephalitis, this means there's an inflammation of the brain parenchyma. The brain itself is inflamed. But when we say meningitis, it means that there's an inflammation of the meninges, the layers that we just discussed. For the meningitis, the diagnosis is made by a lumbar tap, which will be discussed in further sections. Now, moving to the important topic here, which is the CSF, the cerebrospinal fluid. We said it is found in the subarachnoid space, but what is it? Now, the CSF is a clear and colorless fluid derived from blood plasma. It looks like water. The functions of the CSF is to provide a mechanical support to the CNS from blows and trauma. يعني as if you brought here the brain and dip it into a bowl of water. What will happen? In this way, you will reduce the weight of the brain and you will protect it. So if it is, for example, if any blow or trauma happens to the brain, the damage will be much less because it is surrounded by this fluid. Okay, this fluid also can nourish the brain, and also it contains some biochemical compounds like neuromodulators that are important also for the functioning of the brain. So we said it's derived from blood plasma, right? So is it the same composition exactly? No, of course not. There will be some differences. So what's the difference? The CSF has more sodium, chloride, and hydrogen. However, the blood plasma has more proteins, calcium, and potassium. Yani, you can think of it as, wallah, the CSF is slightly salty, so it has more sodium chloride or hydrogen, and so on. Okay, now in adults, the total, this is an information just to know, yani, for your information, the total CSF volume of about 150 milliliters, like half a cup, is replaced every 8 hours. Yani you're washing your brain every 8 hours by 150 milliliters. And daily you have a total of 100 milliliters of CSF formed. So how is CSF formed? The CSF formed by a plexus or a structure called the choroid plexus. I will show you the choroid plexus first, just to imagine. Here's the choroid plexus. Now, you have to know that the brain, yes, it is a solid organ, but it also has some spaces within it. These spaces are called ventricles, okay? So within these ventricles, we have these choroid plexuses. So what is a choroid plexus? A choroid plexus is simply, I will show you the picture first. This is a zoom in the choroid plexus. It is simply a capillary, okay, that has endothelial cells, and this capillary is surrounded by pia mater and basal membrane, and outside the capillary we can see the epithelial cells of the choroid plexus, which are called the ependymal cells. So again, the choroid plexus, the whole choroid plexus, is composed of three important structures. First of all, the capillary with endothelial cells and the basement membrane, and the epithelial cells, which are the ependymal cells. The same name. Endothelial cells because these are inside, and epithelial cells because they are outside, and we also call them ependymal cells. So, the choroid plexus that hang from the roof of each ventricle form CSF, and these plexuses are font-shaped clusters of broad, thin-walled capillaries, enclosed first by pia mater and then by a layer of ependymal cells lining the ventricles. We saw them. Now these capillaries are fairly permeable. I will show you again. We said this is the capillary with the endothelial cells. Now these endothelial cells have some fenestrations in between. Fenestrations yani spaces. Okay, so these spaces make the capillary permeable. So they can, molecules here and ions can diffuse easily through the capillary because of these empty spaces, the fenestrations. However, if you can see here, within the ependymal cells, we have strong, tight junctions between the cells. These tight junctions are nearly impermeable. They have very selective permeability. Okay, this is important, by the way. We will mention why. So, the capillaries are fairly permeable, and tissue fluid filters continuously from the bloodstream. However, the choroid plexus ependymal cells are joined by tight junctions, and they have ion pumps that allow them to modify this filtrate by actively transporting only certain ions across their membranes into the CSF pool. What does that mean? We said we said that the CSF is derived from blood plasma, right? Okay, so this is the blood plasma. I'll change the color. This is the blood plasma. The blood plasma will diffuse through the capillary as it is permeable, and then it reaches here through the ependymal cells. But because we have these tight junctions, so only specific ions and molecules will diffuse through the CSF pool, and within the cavity of the ventricle. So we have specialized pumps here that will allow specific ions to diffuse. So CSF forms as a filtrate containing glucose. We need glucose, we need oxygen, vitamins, and ions like sodium, chloride, magnesium, etc. So these ions and molecules will be filtrating from the blood to the CSF. At the same time, we also have a diffusion from the CSF to the blood. Diffusion of what? Wastes, unnecessary solutes, they are all absorbed back to the blood. We always want to maintain a good ion and solutes concentration within the CSF, because here in the CSF we have neurons, so we don't want to expose the neurons to unwanted or unnecessary substances. We want to stabilize the environment so that neurons are kept healthy. Okay, so any wastes will be going back to the blood, and only necessary ions and molecules will diffuse from the blood through the ependymal cells to the CSF pool. Okay, so the careful regulation of CSF composition is important because, as we said, CSF mixes with extracellular fluid, bathing neurons, and influences the composition of this fluid. Now, ion pumping also sets up ionic gradient that causes water to diffuse into the ventricles. What does that mean? Now, we said that we are pumping ions and molecules from the blood to the CSF, right? Now, a rule to remember: water loves solutes. So wherever there is solutes, water will diffuse. Whenever we have a high composition of solutes in a space, water will go to that space to dilute it. So because we have this ionic gradient, because ions diffused from the blood to the CSF pool, water will also follow and will be there within the cavity of the ventricle. Okay. Now lastly, the choroid plexus also helps cleanse the CSF by removing waste products and unnecessary solutes, as we mentioned. This is the choroid plexus. It is also called, by the way, a blood CSF barrier, because here is the blood, and here is the CSF, and this is the whole barrier. So it's composed of the endothelial cells, the basal membrane, and the ependymal cells. This is the barrier. Now, after it is formed, it moves from the ventricle through the spaces within the brain. Here, for example, it is here, for example, in the third ventricle. This is called the third ventricle. This is a space within the brain. The CSF will be formed here. Then it will move through the spaces. It reaches here. This is another space within the brain called the fourth ventricle. The fourth ventricle has two openings or apertures. These are the lateral aperture and the median aperture. Just know the names for now. These apertures or openings will allow the CSF to diffuse or to move from these ventricles to the subarachnoid space. Remember, now we said the subarachnoid space is the space where the CSF is found. So they will move here from the ventricles to the subarachnoid space, as you can see here. And once it reaches the subarachnoid space, it gets into the arachnoid granules that will drain the CSF into the sagittal sinus here, the superior sagittal sinus. So remember, I told you the arachnoid granulations have some function. So this is their function. They absorb the CSF into the venous sinuses, the superior venous or the superior sagittal sinus. So this is the circulation of the CSF, starting from the choroid plexus in the ventricles through the lateral and median apertures of the fourth ventricle into the subarachnoid space through the arachnoid granulations and into the venous sinuses or the superior sagittal sinus. This superior sagittal sinus and other venous sinuses within the brain will eventually drain into the internal jugular vein, which is found in the neck. Now, a good clinical correlation here is a case called hydrocephalus. Hydrocephalus is water on the brain. Like here what happens is that there's an increased amount of the CSF within the cranial cavity. Now, what will be the cause? It could be either an increase in the production of CSF, or there is an obstruction in the outflow of the CSF. For example, it could be an obstruction in the ventricle here, for example, or an obstruction here within the arachnoid granulation. It could be due to inflammation of the meninges leading to obstruction. So all these will cause accumulation of CSF within the brain. Now, the case here will be different between adults and infants. Why? In infants, see here, this is the skull of the infants. The sutures are not yet closed in infants. They have the capability, the skull has the capability to expand, to enlarge. So if there is an accumulation of fluid within the brain, the skull will enlarge to allow for the accommodation of the high fluid. So this will cause a condition called hydrocephalus. However, in adults, because our sutures are already fixed, there's no space to enlarge. So when the CSF accumulates, it will cause compression on the brain, and it might be fatal. So what do we do in this case? We place a tube or a shunt to try to relieve the pressure by allowing the CSF to drain from the ventricles to the abdominal cavity or the cardiovascular system. Now, moving to a very important topic, which is the blood-brain barrier. The blood-brain barrier is a protective mechanism that helps maintain the brain's stable environment. Now, what is the blood-brain barrier components? First of all, we have here a capillary. This capillary is having endothelial cells, and these endothelial cells are held by tight junctions. Outside the capillary, we have a basement membrane, and outside the basement membrane, we have pericytes. These are pericytes, the smooth muscle-like cells, and we have the food processes of the astrocytes. So astrocytes are here. It's a type of cells, neuronal cells within the brain. Their food processes surround the capillary. So this is the blood-brain barrier. Now, why is it important? Because we want to prevent the brain from the molecules within the blood. We don't want all the molecules within the blood to reach the brain, because this may cause disruption. And for example, exposure to chemical variations like some hormones, amino acids, neurotransmitters, some ions like potassium will modify the neuronal firing, the threshold for neuronal firing. So if we have excessive neurotransmitters or ions within the brain, the neurons will be firing uncontrollably, and this will lead to damage. So we want a good barrier between the blood and the brain to control the neuronal function. So this is the blood-brain barrier. So again, it has four components: the endothelial cells with the tight junctions, and this is the most important structure or component. Then we have the basement membrane, then we have the pericytes and the astrocytes. Now each one of them has its own function. For the endothelial cells and the tight junctions, as you can see, because they are tight, they allow only for the passage of certain ions and molecules. So what are these ions and molecules? We have two types of transportation. We have simple diffusion. Simple diffusion means that the molecules will be diffusing easily through the tight junctions without any help. Which molecules are allowed for simple diffusion? Lipid-soluble molecules or substances, which are fats, fatty acids, oxygen, and carbon dioxide. and fat-soluble drugs like alcohol, nicotine, and anesthetics. They all can diffuse easily without help. Another mechanism of transportation is the facilitated diffusion. This means that some important molecules will also pass through the tight junctions, but with the help of a pump or a transporter. This transporter will facilitate the diffusion of these nutrients, which are the glucose, the amino acids, and some specific ions. So these are important nutrients that we need in the brain, but we need a pump to facilitate their diffusion. So here there will be a diffusion of lipid-soluble components, and through a pump there will be facilitated diffusion of glucose, amino acids, and some ions. Also, some amino acids, the non-essential amino acids or the potassium ions, will be pumped actively from the brain to the blood, and we don't want them in the brain. So what happens here, and we have here a pump through the endothelial cells or through the blood-brain barrier that will allow for active pumping of potassium ions and non-essential amino acids from the brain to the capillary blood. Okay, across the capillary endothelium. Also, any blood-borne metabolic wastes, proteins, certain toxins, and most drugs are denied entry. So we only allow for lipid-soluble substances and some important nutrients to pass to the brain. The basement membrane. So now we know the function of the endothelial layer and the tight junctions for the transportation of molecules. Then we will move to the basement membrane. The basement membrane has also a function, which is the production of some enzymes that destroy certain chemicals like epinephrine and norepinephrine that would otherwise activate brain neurons. So again, we don't want uncontrollable activation, so we will destroy them by the plasma or the basement membrane. The fourth structure, or the third and fourth, which are the paracytes and the astrocyte feet, what they do is that they signal the endothelial cells to form tight junctions that are very tight. So they will signal for tight junctions formation. So these are the functions of each of the blood-brain barrier components. Now, some areas in the brain lack blood-brain barrier. These are three important areas. You have to remember them. We have the third and the fourth ventricles. Why? Because we saw the third and fourth ventricles here. This is the third and the fourth ventricles. We already have the choroid plexus here, right? Which is the blood CSF barrier, as we mentioned before. So we don't need a blood-brain barrier here in the third and fourth ventricle. So this is one. Also, we have the vomiting center of the brain stem. So first, I have third and the fourth ventricle, and second, I have the vomiting center. Now, why is this important? Because within the vomiting center, I need to sense for any poisonous substances in the blood. I need this sensation of poisonous substances. So I don't want a limitation of sensation. I don't want this blood-brain barrier. No, I want to sense now. So this will be absent here in the vomiting center. Okay? This is important. Again, the last one is the hypothalamus. The hypothalamus also lacks blood-brain barrier. Why? Because the hypothalamus plays a very important role in the regulation of the metabolic activities of our body, like regulation of water balance, the body temperature. So we need the hypothalamus to be also sensitive, to sense for any chemical compositions within the blood. That's why we don't need a limit here. We don't need a limit like the blood-brain barrier. So it's important that the hypothalamus lacks the blood-brain barrier to allow the hypothalamus to sample the chemical composition of the blood and act accordingly. The barrier is also incomplete in newborns and premature infants. That's why they are potentially more prone to toxic substances and infections within the CNS. And usually, an injury to the brain will cause breakdown that reflects changes within the capillary endothelial cells and tight junctions. Remember, we said the blood brain barrier, the most important component here is the endothelial cells and the tight junctions. So if they are damaged, the whole blood-brain barrier will be damaged. And usually when injury happens, it will damage this area, endothelial cells and the tight junctions. Okay, this is a small summary that I did for the blood CSF barrier, اللي هو الchoroid plexus, and the blood-brain barrier. So as a quick recap, this is the blood CSF barrier. It has a capillary with endothelial cells that have fenestrations, so they are permeable, a basal membrane, and an epithelial cell of choroid plexus called ependymal cells that are attached by tight junctions. And here within the blood-brain barrier, we have a capillary with endothelial cells tightened by the tight junctions. Unlike this one, which is fenestrations, here we have tight junctions within the endothelium. Then we have the basement membrane and pericytes and the astrocyte feet. So that's it for our lecture. I hope it was clear. If you have any questions, you can reach us at any time. And thank you for watching.